Hello, everyone. Welcome to today's live broadcast, Why We Should Clinically Classify Genotypes, Not Variants. My name is Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. We'd also like to acknowledge the C&EN Media Group for help with programming and promotion of the event. We have a few important announcements before we begin. This webcast is designed to be interactive. We encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing the presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button at lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Nathaniel Pearson. Nathan Pearson is a Senior Director of Scientific Engagement and Public Outreach at the New York Genome Center, where he and his colleagues build simple, engaging tools that compare one whole human genome to the next in order to cast new light on human health and history. Today, working with a talented team of scientists and programmers, he helps researchers and consumers sift through the billions of letters in each human genome and understand which important spellings make each person unique. If you would like more information about our speaker, please click on his photo image in the top right. The planners for this event have stated no relevant financial relationships. However, Dr. Kevin Davies did disclose that he has a scientific advisory board member, serves as one for Pathway Genomics, and is on the Speakers Bureau for Harry Walker Incorporated, and that neither constitute a conflict of interest. Please note that Dr. Pearson has indicated he has no relevant financial relationships relative to this topic. We'll now turn it over to Dr. Pearson for his presentation.
Thank you, Judy. And just confirming everybody can hear me. So welcome everybody around the planet. It's really, really great to be talking with folks today, grappling with precision medicine, hopefully turning medicine more precise instead of vague medicine. And absolutely tickled and honored to be speaking today among other speakers who have already graced the virtual podium here, whom you may have heard of and heard today. Just a wonderful event. So depending on where you are on the planet, or if astronaut Scott Kelly is tuning in from above the planet, your weather may vary, but here in New York it's really dreary this afternoon. But I want us all, regardless of the weather on our part of the planet, to do a little thought experiment and imagine going on a spring walk on a nice day, say with your niece, a niece of yours who happens to be a young naturalist. And it's a long walk, and your niece gets tired from chasing rabbits around, very curious about the creatures that she's seeing. And after the walk, you go to a drugstore to get her something to drink, a bottle of something that'll quench the thirst of just having chased rabbits all day. And I want to talk about what you might encounter at a drugstore and what you might not encounter at a drugstore as it relates eventually to personalized medicine. So imagine that you go to a drugstore and you see a bottle like this. And it's got a pitch on it, an ad that says, "Drink me," tells you how much is there, and it's got a little label that gets at some ingredients. And this, I think, is a crux I want you to pay attention to. We're going to talk today about genome labeling, about truth in what's in the bottle and what it means to us in terms of health and how we explain that. So imagine that the label on the bottle said contains carbon, hydrogen, nitrogen, and oxygen. And each one of those elements was listed and starred with a little caveat that said maybe hazardous. And I want to point this out that this is not what we see when we go to a drugstore today. Of course, we see things that are labeled differently. But in doing so, I want to point out through this analogy that I'll get to, that we're doing this in genomics today. We're doing this the wrong way. We're labeling things wrong. And today I'm going to talk about how we can label them more precisely to better understand what's going on and actually make medicine more personalized. So imagine that instead of a bottle of a liquid for quenching thirst, we have a bottle of genomic information, a genome from a person. So 6.6 gigabases, quite a flagon of information to deal with. Murky stuff inside in terms of what it may contain and what may harm us, what may not. And that's what we'd like to understand, is that label that we get back, be as a physician, a patient, a family member of a patient, a payer, whomever it may be, whatever stakeholder we are in the system here. Imagine we unscroll what's inside, and we might find some interesting content. So these are variants from my own genome that when I read my whole genome, these are some of the ones that I was quite curious. So there's a couple of interesting carrier variants in here in DLD and ACADS. And there are some novel nonsense variants, or not, in some cases quite rare nonsense variants, not always novel, but variants that cut short proteins and that make me curious about whether or not they may affect my health in a particular way. And the question is, how do we interpret these variants that inevitably we find in any person's genome? So I want to liken this question to that faced by a previous naturalist. And of course, you may have recognized the naturalist chasing rabbits as dating back to 150 years ago. Alice chasing a rabbit into Wonderland. And of course, one of the great characters in that story asked about rare nonsense, about uncommon nonsense, and said, I should like to have it explained. So the Mock Turtle's question here, uncommon nonsense, I should like to have it explained, rang a bell for me as often what we're trying to do and interpret a given human genome today, understand that rare variant that may look intriguing to us functionally, but we don't quite know how to interpret it. So if we cast our time machine back to that year, 1865, of course, and we had, say, a panel of TED speakers, many of the famous ones would be dead white men today. Charles Dodgson, the writer of Alice in Wonderland, might be one of the speakers there. There might be Charles Darwin, who at that point was working, I think, on the French translation of On the Origin of Species, having sort of like launched his great work into the world and have it kind of pervade science already. Nearby, but alas, not knowing Darwin, we had Gregor Mendel finishing up the last tally of a very crucial crop of peas that was going to introduce us into the basis of systematically probing genetics and the heritability of phenotypes. And a fourth speaker whom we might not have heard of now might be a guy named de Champs-Courtois. And I want to introduce him. If you don't know who de Champs-Courtois is, he actually figures quite prominently in the early history of chemistry. And I think this is where I'm tickled that Kevin Davis invited me, because Kevin has embarked on a new career that involves in some ways helping explain chemistry to the world in addition to genomics. But if we cast back to that year, de Champs-Courtois brought us the first helix. It wasn't a double helix, the first helix in chemistry. And this was a predecessor of the periodic table. So de Champs-Courtois noticed that the properties of elements varied in a periodic way, in a recurring way, by their molecular weight. And he made this spiral, this helix model called the telluric helix to help us understand that periodicity. And this was a real important forerunner of Mendeleev's later periodic table. So if we think back to our understanding of chemistry at that time, let's do a little history lesson here. What do we know about chemistry and health? So back in 1865, we knew about 56 elements, being the simplest stuff that we can easily isolate on the planet. De Champs-Courtois figured prominently in cataloging those, noticing that their properties varied in a periodic way by weight, and that those properties in turn were key to understanding the world itself, its composition and the composition of stuff in it, and eventually to the ways we want to tinker with it. So to understanding what is going to blow up in a chemistry lab and what isn't, and what is going to hurt us and what isn't. In addition to elements, we already knew, in addition to those 56 elements, and you might think of the 56 ACMG genes today, but in addition to the elements, we also knew and classified another concept, which were molecules. And molecules specifically being combinations of one or more elements in particular amounts and link configurations. And think about that for a moment, because we're going to see a direct analogy to where we are in genomics. Importantly, of course, molecules do much more than the sum of their constituent elements. So the elements may have particular various properties, carbon, hydrogen, nitrogen, oxygen, but their combination in a molecule in particular amounts and linkages yield very different properties depending on what those amounts and linkages are. And they're more than just the sums of the stoichiometry of that element. Some of them, in fact, we had already figured out their effects on health. So the first toxicological report attributing sort of toxicologically solving a murder traced it to nicotine poisoning. So we already knew that nicotine was part of some bad stuff for us at the time. And so we understood something about the structure of some molecules then. Benzene structure was solved quite early in the 1850s, and about their health effects on us, as distinct from those of the elements that made them up. And importantly, when you learned about these things, you learned about them in grade school. So they were fundamental to every basic education pretty much around the planet today, these two very distinct kinds of concept, and importantly, the complexity of molecules over that of the simplicity of elements. So if we think about that complexity, let's take a very simple chemical formula, C10H14O, and look at some of the molecules that have that formula and their various properties. And some of them figure in famous rock songs, or as a substitute, something you might have gotten in the 60s if had you bought your bag from the wrong dealer. You might get oregano instead. So oil of oregano fits that. Parsley, sage, rosemary, and thyme. So rosemary oil also is the same formula. Thyme oil is a different conformation of the same oil. They all smell slightly different from each other. And then you might think of the Donovan song Saffron. Its wonderful smell is a very different conformation of the same stoichiometry. Pennyroyal, moving along to Nirvana in the 90s, has some health effects beyond just smelling good. It's a mint, but it's been used for a long time as folk medicine to induce abortions, and it's actually unclear how well this works. We might think of other molecules with the same formula, like this one, which is actually from a tree called the headache tree, and you do not want to ingest this one. It actually will make you puke. So the properties of all these vary greatly, and some of them are even used outside of nice-smelling perfumes, but for things like controlling pests. So this is a beetle pheromone that fits the same stoichiometry, but very different properties as the other. And some of them, we even get to complexities like mirror images. So here are two enantiomers with this same formula. The one chirality, one-handedness of the molecule smells like spearmint. The other, exact same formula, just the mirror image, just the flip molecule, smells like caraway. So I think a cool example of how a given molecule is more than the sum of just its constitutive elements in terms of its structure matters and the linkages among those elements matter. So how we don't track the chemistry of health. So knowing that molecules matter in health and that they matter for labeling, for example, when we go to the drugstore, we don't expect pharmacists to keep behind the counter a periodic table where they mark up nitrogen with some background information saying, oh, it's harmless, it's benign in its free form, but if you combine it with a gas and bond it with two O's together, it becomes caustic as a caustic gas. Or if you make it with a triple bond to a nitrogen, turning it into cyanide, it turns acutely toxic. Or it's neurotoxic in some alkaline combinations with CH and O. Plenty of alkaloid drugs, et cetera, have that particular composition. This is not how we track the chemistry of health through like a heavily marked up periodic table of elements. So we'd end up with something that looks like this. Back to your rock and roll, the periodic table on a Rolling Stones album. This is not how it's done in a pharmacy today. Rather, a pharmacist or a pharmacologist, in understanding what's inside a bottle and how it may affect you, is going to go to a database of information about molecules, not about elements themselves. So we might track the properties of a molecule like methamphetamine in terms of health in a database that's dedicated to that, building on maybe you saw these in med school if you're a physician, the old Beilstein German language catalog of organic chemistry, but more modern versions like PubChem that now catalog this knowledge systematically, outside of the concept of element, but in that of compound. And you might think of the labeling question. So if we labeled a nice healthy food like a banana this way, we might actually label it by the molecules it contains. And here's a great example of what we'd see if we labeled something we consider natural. By contrast, how are we tracking the genomics of health? So when we open up that genome bottle and try to drink from it and get some insight, how are we understanding what's inside first? Conventionally, we're tracking a very simple concept, that is variance. So variance being the simplest way that two chromosomes can heritably differ. So you can imagine a simple spelling change. They're often just a single base substitution. They could be a bit more complex than that we get into long variants and indels, transpositions, inversions, etc. But basically, variants are the atomic way of that chromosomes can differ from each other heritably. Importantly, they may match or mismatch an arbitrary reference genome today. So we can talk about that a bit in questions if we want to. That's an important point. That reference genome brings some biases to the table that we could discuss. And it is really important to understand variants. So their actual spellings in terms of base composition, so it's that C versus that T, or that inversion or that transposition, tell us a lot about the history of the chromosomes involved, so how they've evolved. In turn, the history of the population of organisms that they belong to, so us, how big the population has been, who has migrated with whom and who stood to whom over time, and in turn about physiology, so not just mutation processes and evolutionary history, but what correlates of those may say something about physiology as well. So if we see nonsense variants tend to, for example, cut protein short, and in doing so be more important than missense variants. So that matters, and we should track that knowledge. However, tying individual variants to health typically presumes that those variants have simple, strong, and independent effects. And if you think about this, this is a habit that traces back to our early efforts, really, yeoman efforts in early clinical genetics, where we could only read a few genes at a time, typically in a few people at a time. It was so expensive, and so we'd start with the sickest people first and look at them. We ended up with ascertainment biases, and we ended up with problems later when we looked at more people, many of them more healthy, and started to see that the variants we presumed were harmful, and strongly so, often turned out to be impenetrant. Their effects were weak, or they disappeared on broader statistical survey. And of course, now with the boon of fast, cheap sequencing, we can read many, many more genomes more comprehensively. We're starting to grapple with this problem of impenetrance much more so. And we want to make genomes not just diagnostically useful for people who are urgently sick today or for a tumor, but prognostically useful so that we know what will happen with somebody born with a particular, that genome bottle. So I'm going to argue today that we should instead be clinically classifying and reporting genotypes, not variants themselves. And so we'll go through what this means, but if you have no time, this is the gist of the talk, so you know my main argument here, and the rest will be commentary, and I'll explain why we should do so. But a genotype being a combination of one or more variants at one or more sites on one or more chromosome copies. So it could start as simply as a single site, and you could have a genotype that was C/C, 2 copies of C, CT heterozygote, or 2 copies of T. That would be a genotype. And it elastically can grow to include more sites on that chromosome, of course, but also sites on other chromosomes. So it's a very elastic concept that takes into account something very simple today, a single site, but also multiple sites as our knowledge grows. The variants involved can importantly, in a genotype, as in a molecule, they can interact with each other non-additively, and we're going to show some examples of that importantly, because when I say non-additively, I mean that the genotype's qualities, its properties, will be more than the sum of its constitutive variants. And importantly, if we start to do this, if we start to actually classify them and clinically report genotypes rather than variants, I'm arguing that it puts us in a much more robust track to track our growing insights into the genomic basis of health, along with other factors that matter in health, that starts with today's fairly simple single site in a genome knowledge, single variant focused knowledge, but grows elastically over time as we sequence and look at more people over time and notice the correlates between particular genotypes and particular phenotypes over time. Crucially, I want to make one point here, which is that this is not a new insight. Mendel, from the very start, actually understood this, that we need to be reporting genotypes, and we'll get to that in a moment. So the analogy does extend, and I want to make it here. So if you think of variants as being roughly analogous to elements, the sort of simple atomic way in which things vary in the domain we're looking at, you could imagine that very rare, what we call a rare and penetrant variant, is a bit like a really rare and harmful element, so like something like polonium, where it doesn't really matter if you're looking at polonium chloride or polonium bromide, any salt of polonium is going to be bad for you. You don't want to eat it. And likewise, there are plenty of variants out there that regardless of the genomic background, regardless how complex the rest of that person's genotype is, they are going to harm that person in some way. And those were the ones that we discovered early on in clinical genomics that have turned out to be the most robust findings from early clinical genetics. And I'm arguing here that has biased us towards this sort of myopic view of classifying and reporting variants, thinking that it was always going to be so simple. And it's not so simple. and we'll get to examples. But if we extend the analogy, you could think of reference variants, the ones that we often omit from a report, as being something like the hydrogen that we might omit from a chemical formula because it's given how hydrogen bonds to carbons. So that's sort of often just left out for simplicity and for compactness. There are some variants that recur in the world, in tumors, for example, that happen by new mutation in parallel over and over again, and we can start to look for, and they often are important in health. Those you might think of something like a radionuclide that is often made anew from another element in the world as it decays. And likewise, if we think then of genotypes as being analogous to molecules, that is combinations of one or more of the atomic units, in particular amounts and linkages, then you could imagine asking about a partial genotype. So part of a molecule we might call a moiety, and if it does something important, we might call that a functional group, a hydroxyl group, etc. We will likely start to discover partial genotypes that say enough on their own to be interpreted regardless of the broader genotype that they're part of. And the analogy can extend further. So if you think about the copy, how many copies of a given variant are in a genome, that's something like the stoichiometry of how many copies of an element are in a compound or a given atom are in that compound. If you think about how genotypes are passed on together, that's something like the way that elements are bonded together, either in a strongly linked way, in a covalent way, or in a looser way. They're in the same person and they might get transmitted together, they might not, which you can think of as a bit like an ionic bond as generations go by. And then there's even some kind of little twist to it. So you could remember that example of the spearmint and the caraway that are mirror image molecules. There are, of course, genotypes that have very different effects depending on whether or not you get them from one parent or another. So they're the same genotype, but epigenetically they're imprinted with distinct effects. So you might think there of Prader-Willi and Angelman syndrome, tracing to extra copies of chromosome 15 or parts of it. And it matters very much whether you get that extra copy from mom or from dad. Finally, how are genotypes made, or variants come together, elements come together through a reaction, and of course there's a replication process that brings new variants together. Recombination does that as we reproduce. So there's an extended analogy here where Mendel does mean Mendeleev, and I want to make this the crux of our discussion today. As I noted, Mendel got this from the very start of genomics. He saw this complexity. So if you recall back to his main experiments, he was looking at pea plants, and he was say crossing purple flowered and white flowered pea plants, and he might have expected, or Darwin might have expected at the time, blending, so that the offspring would be, say, pink or lavender flowered. That's of course not what he got. So what he found was that the offspring, the kids of these pea plants, remained either purple flowered or white flowered. And from the clever math that he did, he deduced that this traced to effectively a genotype, and importantly, a non-additive genotype. So if inheritance in Mendel's world were additive, then these heterozygote blooms would have been pink. Instead, they are purple because of the dominance of the big R allele, the purple allele, over the little R allele. And that dominance turned out to be so widespread and so easily found in genomics that we've actually forgotten that it's just non-additive genomic interaction at the genotype level. We gave it a special name, and we've tracked it in our clinical databases as dominant versus recessive forms of disease, and in some cases we get tripped up. We're looking for one and we find the other in a particular patient. But we tend to track this as sort of like a flag, as an add-on in how we report variants, rather than as something that could be more neatly subsumed into a broader understanding of non-additive genotype reporting. There are other examples of non-additivity that we've known about for a long time. So another might be sex chromosome genotype dependence, and this is one that's sort of so obvious that we've forgotten that it actually is a genomic interaction and a non-additive one. So if you are a physician and you get a report for somebody with a particular BRCA1 or 2 variant that looks scary from the literature, it matters very much whether or not that person has an XX genotype or XY. Now, of course, anybody can get breast cancer, so men get breast cancer too. Nonetheless, the risks are much higher on an XX genomic background than an XY genomic background in that particular case. And again, this is one that's sort of so obvious, we've often forgot about it, that it does reflect a non-additive genomic interaction that we already understand. There are other examples that we've sort of run across and started to grapple with. and we've often sort of swept them under their own special category rugs. So mitochondrial variation, because we only get one copy of our mitochondrial genome from one parent, and it is that genome doesn't recombine, but rather accumulates variation over time in a haplotype setting. We tend to think of the effects of haplotypes in mitochondrial variation, where we've grappled with that before we have elsewhere in our genomes. And there are a few other places where we have long-standing haplotype diversity that's stable thanks to various processes evolutionarily, such as HLA and immunoglobulin loci and KIR loci, these other hypervariable immune-relevant parts of our genomes, as well as long haplotypes that are relevant to drug response, such as the enzymes that encode the genes that encode many liver enzymes for detoxifying drugs, the CYP450 genes. Those we've started to record our functional knowledge, typically not at the simple variant level, but often at the haplotype level. We've started to see that this stuff is a bit more complex than reporting single variants. Now, let's talk about some other emerging insights into non-additivity that have not been understood for so long, but will shine the light on where this will go as we look at more and more people's genomes and want to make our understanding of their long-term health arc more precise. So we can start it within the same gene. We've understood compound etiology for a while. We often, when we're looking at a sick child and what's making her sick, we may find that case of disease traces to distinct variants in the same gene that would each nominally be recessive. So normally you need 2 copies to get sick, but effectively they turn each other dominant. And that's what we call compound het etiology. There are many examples in the literature. Many of us have started to report those. If we're clinical geneticists, for empowered patient families, we may have to consider that possibility in looking at what's making a child sick in the family. There are also emerging new examples where what's really interesting is that the rare genotype is actually associated not with disease but with health. So this is a great example from the prion protein. So the gene that encodes the prion protein is PRP1. I encourage you to look at this particular article where this is a really cool example because there is a dimorphism. There's a simple one base substitution in PRP in the 129th codon, where some copies of that gene encode a methionine there, and some copies encode a valine. Actually, both of those variants might get called pathogenic because it turns out, oddly, that homozygotes they tend to get prion. They are less susceptible to prion disease than heterozygotes. So if you have 2 copies of an M there, I'm sorry, I said that wrong. So either if you have 2 copies of an M or if you have 2 copies of a V, you are susceptible to prion disease, whereas if hets are less susceptible. So, but either variant there can effectively be called harmful with respect to prion susceptibility. Nonetheless, in a study of folks from New Guinea, where people have eaten brain tissue for a long time, including from people, it was an intriguing new variant found very nearby in that gene that actually appears to block prion disease completely. So at the 127th codon in that gene, if you have this particular rare variant, you appear to be, so far, immune to prion disease, utterly immune, regardless of what genotype you have nearby at codon 129. So this would be a great example of emerging insights from looking at many people in the world, including previously understudied people on populations on the planet, of finding rare variation that makes a rare genotype that is actually health, that drives health rather than disease. So further examples, not just within the same gene, but across genes. So this is a great example of so-called digenic etiology, where, again, we have variants that would normally be called recessive. So a variant in RDS or in ROM1, you normally need 2 copies of either one to get retinitis pigmentosa. This is you can look at this old paper. It's fairly well known now. But effectively, they turn each other dominant. This is like compound het across genes, where if you happen to get both, you tend to get RP, and it's bad news. But this is a non-additive interaction across genes. And likewise, we can flip that and think about rare gene type associated health across genes as well. So a recent study at population scale in Iceland found an intriguing new variant in APP that as part of any genotype background that folks have looked at, including ApoE4, basically buffers the expected pathogenic effect of ApoE4. So ApoE4, people who carry an ApoE4 allele tend to, if they're hets, they tend to have a modestly higher incidence of Alzheimer's disease. If they're homozygous for that particular variant, they tend to have much higher. But if they carry this particular rare variant in APP, in addition, that genotype together is there's no difference. It's a fairly low risk for Alzheimer's disease regardless. So again, we need to be looking at the person's genotype as a whole in terms of what's already known about it to understand their outlook for AD in this case, not just at the single site in ApoE that we would typically look at. And this gets even murkier when we come to the ACMG 56 genes themselves. So famously, of course, ACMG took a look at what we already know about the genetic basis of important adult onset diseases that we could do something about. And they said, what genes do we know most about their roles in adult onset diseases where there's some screening or prevention and or treatment option available to help someone? And they made a list of 56 genes. It's a really great first start at knowing what kinds of things we would like to report, especially as incidental findings from looking at someone's genome. One of the genes, or actually two of the genes on that list are MLH1 and MSH2. Both of those genes are strongly implicated in colorectal cancer, so in Lynch syndrome and other colorectal cancers. Nonetheless, this study was a really important one that I'd point you to, showing how little we yet understand about the long-term risk of that disease, even from this best understood of genes. So the study looked at a couple hundred families, in each family there was a pathogenic variant, a nominally by ClinVar annotated pathogenic variant segregating in that family. And they looked at whether or not folks who had a copy of that variant actually got colorectal cancer by a particular age. I think they were looking at men in the families by getting cancer by 70. They found in about a sixth of the families that 9 of 10 men or more would get colorectal cancer by 70. So that's effectively what we want. We don't want people to get cancer, of course, but we want to be able to predict from a single variant that they likely will, and then hopefully do something about it. However, the sobering flip side is that we found in another sixth of the families that the lifetime risk appeared to be less than 10%. So these are the same variants, different families, different genomic backgrounds, different lifestyles, et cetera. There are other interactions at play here that are going to help explain, hopefully over time, why we yet know so little, even for this well understood ACMG 56 listed gene, why we understand so little about whether or not someone will or won't get colorectal cancer given a particular variant. And hopefully, as we get more and more big data from populations, including genomic data, but also the ancillary data that goes with it, and track people over time, we can start to understand some of those interactions. And some of them may turn out to be genetic, and likely in some cases non-additive, the same way we've seen before. So what I'm arguing today is that all of us together need to get our health infrastructure, our health informatic infrastructure in particular, ready to record and convey that knowledge as it emerges from the data that we now have at hand and coming. And why this matters, of course, for healthcare is why we're here today. We want to sharpen healthcare, to make it more precise for us, for our families, for our patients, for everyone in our lives and on the planet. And importantly, that starts with the job of caregivers themselves. So making life easier for a caregiver is one key way in which the innovations that we propose can be taken up. So currently, let's take a look at what, and by the way, a doctor doesn't want a flagon of insight from a genome. They want a little espresso. So they want to be able to sip this espresso on a report, know what to do, and act on it. And we'd like to help them do that. So how do we streamline the job of a doctor with genotypes versus variants? Well, let's take a look at a hypothetical genome today as it might be reported back. This is a particularly unlucky looking one. So this person might have 5 particular variants called as pathogenic, which would be kind of scary. Typically, most of us would have probably 0 to 2 such variants called, but this is a jackpot that gets 5. And so this is a scary list to go through for the patient, but also for the caregiver who's trying to walk through and understand what, if anything, can be done and how to explain these findings to a patient. So the particular variants in question might be implicated in breast cancer, in long QT syndrome, in LDL, in sort of blood lipids, in anemias, and beta thalassemia, for example. So if we bring in today's single-site genotype knowledge, rather than just reporting these variants, the picture changes a fair bit. So now we're looking at the genotypes of this person, not just the variants that got flagged, and we're clinically classifying those genotypes. So we no longer have 5 red ones, 5 pathogenics, but rather we have 3 genotypes that at this simple view would still be deemed pathogenic, and 2 that might be deemed, instead, we might call them family risk. And let's walk through those. So here we're still heterozygous for this variant in a BRCA gene. It looks scary. In a gene implicated in long QT, and in one implicated in high, low-density lipoprotein. Now, but the genotypes for hemolytic anemia, and that's the rhesus genotype, which is a really cool one. It's underdominant. It's the rare case in human genetics where heterozygotes are at a disadvantage, and we could go through that offline. It's a really cool little story. But if you're homozygous, it's more of a family risk question. And likewise, the beta thalassemia risk is, if you're heterozygous, it's more of a family risk question for family members who might be homozygous for that same variant. So now we've made the report a bit more precise in terms of how we classify pathogenic for the person versus for family risk. And we made it a bit easier, I think, for the doctor to go through the findings and know what's most important to talk about first. Now, if we extend further beyond these single sites to incorporate what we already know, so these are already informative genotypes across multiple sites in this person's genome, we can get even sharper. And actually, it turns out that all of these genotypes basically collapse to being just family risk. There is nothing pathogenically alarming among the 5 listed here for the person at hand, but rather they might inform screening for other family members. So, for example, they might be male, they might have an XY genotype that will likely buffer that really strongly affected, strong effect expected for the BRCA variant. Nonetheless, it might mean that they should tell other members of their family eventually, either now or later, that they might want to be screened for that variant as well if they have an XX background in particular. And likewise, based on the papers that we already know about, we could collapse all of these to similar kinds of family screening questions. But at the genotype level, we might find something new as well. So here is a case of a genotype across multiple sites in 2 different genes on 2 different chromosomes in this person, none of the variants in question here would on their own be called pathogenic, but together they confer a quite strong risk for ankylosing spondylitis, but based on what's known about the interaction between one of the HLA genes on chromosome 6 and a gene called ERAP1. And so none of these, the constituent variants of this genotype would rise to the in ClinVar to being called pathogenic, but the combination would be clearly so. So I would argue that this is quite alarming in terms of if you look at the papers and this person's risk for developing AS over time. So the genotype can help us both set folks at ease and make the doctor's job a bit easier for explaining about family screening versus alarmingly having to go through 5 fire alarms for that person. But it can also clue us into new findings from that person that we wouldn't have seen if we were only reporting back single variants, but instead we were looking at what we already know about genotypes. There are some clever twists and some tough twists to this, and I want to acknowledge those, and we're going to have to grapple with them going forward. So we've looked at a lot of people's genomes now. We find that there are tons of variants out there. So we already know about many dozens of millions of simple variants among people's genomes. And it turns out that given how fast the human population has grown recently, the more people we sequence, we can expect to continue to find rare variants in abundance. So variants that are individually rare, but collectively very common and just rife among us. Likewise, if you think about how all those millions of variants could be combined, it's boggling, right? So there are many more possible genotypes yet than variants. They can be combined in almost infinite possible combinations. And so we need infrastructure for being able to track what we know about those. Now, the lucky thing is here that we don't have to track, we don't have to have an entry in our database for every genotype that's possible. We only have to track the ones that we already know are informative in some way. And that list will grow much more slowly than the exponential possibilities. So we only need to track informative genotypes, and in particular clinically informative ones, as they slowly accrue beyond the single site knowledge we have now. But I'm arguing that by converting to our computing systems now to track genotypes this way, including with ambiguity, so knowing that, for example, that this genotype, this part of this genotype could be either het or homozygous, but it has to be one or the other. So we need ambiguity codes built into how we encode genotypes. Even doing so would only roughly double our database size today. Versus if you take a database like ClinVar, which is actually still quite small, it's on the order of a few dozen thousand variants that are clinically classified. If we classified genotypes instead, we would barely double that size. We'd still only have dozens of thousands of genotypes to keep track of that would be clinically classifiable today. And that would grow slowly as we look at more people over time. Genotypes can overlap each other, so they can fully, one genotype can subsume another completely, or they can partially overlap. So we need structured syntax and what informaticians would call schema, database schema, that is well thought out ways of encoding the data in rows and columns, and with keys and values, so crucial IDs that tie one table to another that let us track that overlap in smart ways, so that we know when we know something about a genotype that overlaps another one, we can know whether or not to functionally that the second genotype that may be newly discovered may act in some ways similar to the other one. And that's a bit like knowing whether a newly synthesized molecule is going to act like one that it shares some moieties with, for example. This is completely tractable, and of course much more so now that database science has become quite a rigorous and well-structured method in its own right. And so we have good computational methods for doing this in other domains, and we can do this, I think, much better in silico now than we could tracking it on paper in the past. Now, the other twist here that makes looking at genotypes and their health effects tough is that genotypes, a genotype is going to be found in no more people than any of its constitutive variants. So each genotype is going to tend to be rarer than even the rarest variant within it. And this makes detecting associations statistically hard. And we already face this with single variants, that a variant may be in too few people in the world to significantly associate with disease. Even if it's only found in sick people, there may be just too few of them on the planet to know whether that's significant or not. And there, of course, we have to turn as genomicists to things like rare variant burden tests, other kinds of rare variant association tests that pool variants together to ask statistical questions. And we've got some methods for doing that here, and we'd have to adapt those methods for looking at genotypes as well. Importantly, we only come to track them once we have evidence that a given genotype does matter in health. We only need to track it again when it becomes clinically informative across people. And the other twist and wrinkle that arises because genotypes are found each in few people is that they are more informative about the identity of that person. Now, I would argue here that privacy worry presumes that genome data are going to be widespread, that we will be in a world soon where we're subsumed in we're suffused in genome data. And so it would be possible to know that a given someone in a genome database, a genome that was in a database and had a particular genotype, we'd have to have other information about the person who carries it in the world in order to be able to match them together. So we may come to that point, and we'll have to deal with that when we do. But there will be trade-offs there in terms of the clinical utility of this data across labs and helping patients versus the privacy concerns of each of the people who may carry a given genotype. And finally, variants, of course, they still matter. So we still need a periodic table. We haven't jettisoned the periodic table just because we have PubChem. It still matters to catalog what we know about variants. They still tell us about mutation processes in the past, about population history. And we still need to flag them within genotypes, as we've noted, where they might be shared in a different genotype in somebody else in the family where it matters more. So it's on an X background versus XY, et cetera, in the simple case. But in the cases to come, where we learn more about other kinds of interaction, we will need to know that your sibling is much more likely to carry one of your rare variants than your first cousin, and yet more likely, than your second or cousin or a distant stranger. The take-homes that I want us to think about today, and hoping you guys will take home and we can talk about in questions, include that. So I'm basically arguing today that we need to start classifying clinically informative genotypes now, and that doing so will help basically make healthcare more robust, make our infrastructure more robust for its narrow use today, where we use genomes today to diagnose. Right? We have cells growing too fast to wait. So a sick child growing too fast to wait for sequencing to get cheaper or for knowledge to grow, or we have a tumor growing too fast for sequencing to get cheaper or for knowledge to grow. And we want to look at the genome of that tumor or of that sick child today and know if there's any foothold into helping diagnose and treat something urgent. The longer term broad use of genomes that we all aspire to and that the precision medicine efforts are built around is to make genomes prognostically useful, not just urgently diagnostically useful, but useful looking ahead from womb to old age as a part of long-term health management. And that involves the much more murky question of how someone's health is going to slowly evolve over time, not necessarily be already understood and to be diagnosed yet. And I'm arguing here that because genotypes matter in that question of impenetrance, of the crystal ball of whether a given variant is going to make someone sick, that classifying and reporting now, getting people used to working with genotypes, is going to make that much easier for our whole healthcare system when it does become possible. And importantly, the key is to ease caregiver jobs. And this takes me back to a great essay by, I think Atul Gawande wrote a New Yorker article about 3 years ago about the uptake of two revolutionary technologies in the 1800s. So one was hand washing and one was surgical anesthetics. And anesthesia, they were both invented, I think, in the mid-19th century. And one of them was taken up by surgeons overnight, and one of them took 30 years to take up. And of course, anesthesia was the one that was taken up right away. And hand washing was the one that took a long time. And Gawande, he argues that the reason was that both of them made life great for patients. They both extended millions of lives over time, but only one of them made doctors' lives easier. Right? Anesthesia turned surgery from what was basically a carefully orchestrated attack on a patient with a lot of screaming and holding people down and blood and tears to one that was much more the serene, precise art that we think of as surgery today, where there's music playing in the operating theater, etc. Hand washing, by contrast, made life tedious. It meant you had to be very careful about what you did and keep track of even more procedures than you would otherwise have to in the operating room. And so here I'm arguing that classifying genotypes would actually make doctors' lives easier. It would keep the reports shorter and more precise, more clear than what they need to explain to a patient, and less full of the false alarm, five red alarm variants, but rather just a few findings that might matter for that patient. And importantly, all this in the long run can help lengthen lives by conveying insights about people's health going forward more sharply. And this is what the essence of precision medicine. And I argue that we should switch now. So rather than waiting, rather than wringing our hands and saying, well, of course, this sounds great. We should be talking about genotypes, but my lab can wait because we have a database right now of variants, and it's going to be hard to switch over. It's only going to get harder. So our databases right now are fairly small, and they're fairly young still in terms of the computing that we put into them. They're going to grow over time, and they're going to get more deeply embedded in many of the systems that as various as stakeholders in healthcare that we run. And we should switch now to genotype tracking in our computational infrastructure before that variant centrism really, really embeds itself in everything that we do and has sort of tendrils throughout our whole system. So what can you do, depending on who you are, what kind of stakeholder you are? If you're an informatician if your job is to help structure data and get it ready for scientists and clinicians to look at later, structure your data now for multi-site genotypes. Build it so that you can accommodate that elastic concept that starts with a single site now, but could accommodate insights across multiple sites on a chromosome or even on different chromosomes over time. And we can talk offline about how that helps today, even for quality control questions that are crucial in today's short-read genome data. If, by contrast, you're a genomicist, a scientist, or if you happen to be someone who's been sequenced already, share whole genomes, not just flattened allele frequency tables of single variants. That will help today to boost power of studies. It will help us understand whether or not genotypes, particular genotypes, actually correlate with disease more sharply than individual variants may. And so, think in whole genomes and share whole genomes if you can, if you have that chance as a scientist. And if you happen to have been sequenced, share your genome. It's a great thing to do on behalf of science. If you're someone who works at the bench, so not just in a dry lab, but actually at a wet lab, what you can do is help us understand the functional effects of genotypes by actually assaying them in parallel. And this gets at one of the challenges we saw earlier, that if a genotype is really rare in the world, it may be impossible to know statistically how it affects health. But we have emerging ways through CRISPR and other highly parallelizable technologies in the lab to directly assay that functionally. And so you could actually imagine not just tweaking the genomes of a cell line to have a single variant, but actually tweaking them to have a slightly more complex genotype that was found in a given patient, and asking whether or not, under conditions that approximate those that we think drive disease in a person, that the cells in that dish act in some way that reflects real etiology, or whether we're on the wrong lead. If you're a clinical geneticist, if you're interpreting the findings that come from genomes today, think and report genotypes. So again, it's not that much more complex to make that final report to a clinician specify that it's C/T rather than just the T, or specify that it's C/T plus XX. Get folks used to looking at that so that it becomes second nature, and they won't think twice. They will understand, if they need to, the basis of your findings more clearly, and the basis of the recommendations that you pass on in clinical reports about patients and about other folks incidentally sequenced in their families. So maybe the parents who have incidental findings as well. And importantly, if you are that caregiver, if you're a doctor, if you're a nurse practitioner, if you're a patient, think in genotypes and request them. So if your clinical geneticist is not reporting genotypes, and is in that sense leaving more ambiguity in terms of how do I report this set of 3 red flags? Are they all red flags for the person in hand, or is something else known in the literature that would clarify which of these matter most to talk about now with the patient versus to help them understand family screening for? Ask for that. So request genotypes in your reports. Get your clinical geneticist partner used to thinking that way as well. And I'm arguing here that if all of us change our thinking a little bit this way and start hark back to what Mendel already got and to where big data will take us, we can change the culture of how clinical findings are reported from our genomes to be more focused and ready for the complexity that will emerge over time as genotypes of 2, 3, and perhaps even 4 or 5 sites eventually become informative over the course of our lifetimes. So thank you so much for listening to the polemic today, and I'd love to talk with you guys about questions that emerge on your minds now.
Well, thank you for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, the BRAF V600E-like notation does not even indicate zygosity. So it's not a question, it's a comment. Would you want to comment on that?
Yeah, and I think it's classic. So, and often, this particular variant is one of those that recurs in many different tumors, and we'd like to understand it, and especially given that many cancers are already understood as multi-hit diseases, especially in how they respond to treatment. And there are some variants that appear to buffer the effects of other variants in a tumor, or buffer the responses, or exacerbate the responses in non-additive ways. So a great point here, when we get tumor findings on a tumor report, we'd often see something like, yes, It might even say something as simple as this tumor is BRAF positive, omitting anything about the genotype, the variant in question, let alone the genotype of that site. Zygosity does matter, and zygosity matters in even more complex ways in tumors because they actually accumulate copy number variations. So a tumor could start out as heterozygous for V600E. It could get a second copy of the V. It could get a second copy of the E. It could lose a copy of the V and become effectively hemizygous for that. And all that may matter in how it responds to treatment over time. We need to know about that. So oncologists, pathologists, likewise, need to start thinking in genotype terms here, perhaps even more pressingly, because we understand the multi-hit nature of tumor etiology and the role of zygosity and of aneuploidy in tumors themselves.
Several speakers today have this. Oh, I'm sorry. Please.
Can we come back to Kevin's question if we go to Violet? Kevin always gets asked questions.
Anything.
So,
sure.
Yeah, and we'll come back to Kevin if we have time.
Oh, sure. Okay. Wouldn't the database increase quite more than half if you have to keep track of all the non-pathogenic polymorphisms, which can also modulate the effect of pathogenic ones?
So again, I think that we have to have triage for. This is a great question. We have to have triage criteria in mind for what makes a genotype informative. And if the effects of its constitutive of the variants that contribute to it are either purely additive, so if in our statistics they look like they're additive on their own, so they could be reported as effects separately, independently, then we might sweep those into a different category. And there might be an additive modifier that might reduce the overall risk of disease, but not be crucially part of that informative genotype. And that's sort of like knowing what else is in the bottle besides that molecule. We don't incorporate. We might know that yes, there's caffeine in the bottle, not strychnine. Based not just the elements, but we know that there's caffeine in there and not strychnine. We also know that there's a little bit of sugar in the bottle, and its effects are somewhat separate and independent of that of the stuff that makes up the caffeine. And I think that the additive modifier question is a bit like that. If we have non-additive variants that are common but weakly, but we can detect that they actually non-additively matter, then yes, I would argue we should actually incorporate those into the genotype. But I don't think that, given what we know today, that's a particularly common scenario. So I think most of what we know is either fairly clear non-additive interactions, and there are a few examples that we've started to talk about here, or there is sort of long tail, to the best of our understanding, additive weak effects, and those would just be reported separately, if at all, if we even deem them clinically informative at all. In the long, and just to add one more point on Violet's question, in the long run, the database will be more than double its size, but that will be true anyway, even with just variants, right? Because we're going to discover a lot more. So we know our database will more than double in the long run. I'm just arguing that it is tractable today for a lab with computer systems in place to double their data size.
It appears that we have time for just one more question today.
Okay.
So here it is. It would seem that clinically classifying genotypes has significant relevance for gene editing technologies. Can you speak to this relationship?
Yes, and I think that gets at the point 3 here on the last slide. For a bench scientist, what you can do, if you know CRISPR, if you know how to use CRISPR-Cas9 in mice, wow, I mean, parallelizing the functional assay of genotypes that we find could help so much, given that there's only 7 billion people, and maybe we'll have 10 billion if population grows modestly, and that's simply not enough for the statistics involved to know whether some genotypes matter. So the way we're going to have to do that is by directly assaying. And so I'm glad that point got asked, and I see one more question, by the way, about trios. So asking, if we're going to focus on genotypes, shouldn't we always be sequencing parents in the family where they're available, not just the sick kids? And I think that's a great point, Christopher, on making that. Sequencing is getting cheap enough. We can much more sharply understand typically what's going on in terms of the etiology of a sick child by adding the trio information now. So even for the urgent diagnostic use, it matters. And in the long run, for more broadly documenting what genotypes matter, sequencing 3 people and including 2 unrelated is going to help us get there much faster. So thank you for that point.
Thank you. I would once again like to thank Dr. Pearson for his presentation. Do you have any final comments for us today?
So on Kevin's question, what most excites, so Kevin I can't see everything, but I think he's asking what most excites me or any of us here about President Obama's Precision Medicine Initiative. And, you know, this is, hopefully we've talked here a bit about some of the brass tacks that face us and getting beyond the sort of word cloud hype of precision medicine, P4, personalized medicine, whatever, what have we. It's great to have folks at the top in our government understanding that this is a long-term aim, that gathering big genomic data and ancillary data from us as a United States population will help us drive science and make a baby's genome a generation from now much more informative than a typical healthy adult's genome today. I think that's the key, is that when I talk with people, you know, many healthy adults I've talked with looking at their genome, they came in hoping for a crystal ball in health. There is no such crystal ball today. And then there's the so what moment, like, you know, why isn't it more informative? That's the meta lesson. It's not more informative yet for long-term health because we haven't looked at enough people over time in the context of other information, you know, and phenotypes to actually make sense of what a single genome means today. And to get there, we're going to have to do so over a generation. It's going to be a long haul.
Well, thanks again. Today's webcast will be available for on-demand viewing through August of 2016. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. And please join us for the last presentation of the day by Dr. Mwanga from Africa after Dr. Pearson's webinar concludes. Thanks again for joining us. See you next time. Bye.